This is part 2 of the chapter 20 lectures, recorded lectures. We had left off on slide 11 where we are discussing bacteriophages. Remember, bacteriophages are viruses that infect bacteria. They only infect bacteria. Oh gosh, we saw one. Let me go back to it so you can see a picture. Remember the one that looked like a spaceship? This is a bacteriophage, okay? It's a DNA virus, and they undergo two type of kind of stages within their life cycle. One is called the lytic cycle, and during that stage, it's very similar to the one that I went over with you, where basically your virus attaches and penetrates the host cell, and then it tricks the host cell into making multiple copies of the virus. And then when the virus all those new viruses kind of burst open from the cell, it kills the cell. So the host cell undergoes lysis during the lytic cycle. But the lysogenic cycle is when the viral genome is incorporated into the host cell or integrated into the host cell genome. And at some point in time, it may re-enter the lytic cycle, but during the time where it's just dormant, we call this latency. And the latent viral DNA is called a prophage. Diphtheria is a disease that's caused by a prophage-carrying bacterium, which produces a toxin that damages the lining of the upper respiratory tract, restricting breathing. So this is an image of a bacterial cell. You can see this is the bacterium. And here is your bacteriophage that is attached to that bacterial cell. Now the bacterial DNA itself is circular. And then there's the DNA inside of the virus. So the attachment phase is shown here, and then step 2, the penetration. The bacteriophage injects its DNA into the bacterial host cell. The biosynthesis part is when the host cell now uses the genes from the viral DNA to create the components of the virus, which would be the capsid, the green capsid, and then the purple DNA. And then the components are assembled. That's called maturation. And now they're going to burst from the host cell, and that's going to kill the host cell. So that's called the release. This is the lytic cycle. Then the lysogenic cycle happens sometimes after the viral DNA has been injected into the host cell, the penetration stage. Now the viral DNA incorporates itself into the host cell genome. And so the DNA remains dormant. It's called a prophage. That viral DNA is called a prophage, and it remains dormant until there's such time as it is triggered to enter the biosynthesis stage. So enter back into the lytic cycle. And things that can trigger this would be environmental stress, such as low nutrients, low water, not enough shelter, not enough space to grow, just different things like that. Temperature, pH. All right. Now, a retrovirus is another type of virus I want to mention to you. It's the virus that causes AIDS. HIV is a retrovirus. And retroviruses are really, really tricky because what they do is they contain this enzyme called reverse transcriptase. Now, if you remember from Bio 111, transcription is when, let me get my pen. Transcription is when the DNA, which is in the nucleus of the cell, is used to make a copy of itself in the form of messenger RNA. And that messenger RNA takes the DNA code, I'm sorry about all those lines, but it takes the DNA code to the nucleus, to the cytoplasm, to a ribosome, and then translation occurs. That code is translated into a protein. So this process where DNA codes for RNA is called transcription. So reverse transcription is when you use RNA to code for DNA. So reverse transcription uses an enzyme called reverse transcriptase. So this is when RNA codes for, I think that stands for clonal or complementary DNA, but anyway, it's a molecule of DNA. And now that DNA can be used to code for a protein. But the cDNA becomes integrated into the host DNA. Replication occurs as the host DNA replicates. The cDNA is replicated at the same time, simultaneously as the host DNA. And HIV may remain latent or dormant for years. So HIV is a retrovirus, and here is an illustration of the stages that the HIV retrovirus undergoes. First of all, it's an enveloped virus, and it does contain, see the green capsid, and then the RNA that's on the inside. There are 2 strands of RNA inside the capsid, and then there's an envelope. So the spikes on the envelope bind to a receptor on the host cell, that's the attachment, and then the virus enters the host cell by endocytosis, phagocytosis. The host cell phagocytizes the virus, basically, and then reverse transcription occurs, and that is when the viral RNA makes a copy called cDNA, and that cDNA enters the host DNA. It enters the nucleus and combines with the host cell DNA. We call it now a provirus. And then the host cell makes many, many copies of that in the form of viral messenger RNA. And then that is used to generate the components of the virus, the biosynthesis stage. And then the components are assembled, the maturation stage, and then they burst from the cell in the release stage. Okay, so that's the retrovirus. Remember, retroviruses use reverse transcription. Reverse transcriptase is the enzyme involved. Now, flu viruses have 2 spikes. They have an H spike and an N spike. The H spike allows the virus to bind to the receptor on the host cell. There are 16 different types of H spikes. And then the N spike attacks the host cell plasma membrane, and that allows mature viruses to leave the cell. There are 9 different types of N spikes. So it's the type of spikes, it determines the vaccine that has to be made for that flu virus. Now, this is showing you how the human influenza virus, which causes the flu, mutates. And you see the mutations within the H and the N spikes, okay? The N spikes are these orange round ones, and the H spikes are the pointy pink ones. Notice that the H spikes on this virus here have a little yellow stripe, and a pink tip. But after the mutation, then the H spike now has a dark, like a black tip, a modified spike, and that's going to mean a whole new vaccine has to be created to fight that version of the flu. So this second illustration, picture B, is showing you how the bird flu and the human flu virus can kind of, if they come in close contact, such as when humans work with turkeys or chickens, when they work with poultry, in poultry houses, if they come in close contact, when both of them are sick with the flu, then the bird and the human flu can actually kind of mix their DNA. And what can happen then is, notice all the spikes for the bird flu are kind of this bluish purple color, and then the human flu are orange and pink. Now what we have is a combination human-bird flu, where you have the human N spikes and then the bird H spikes. So it's a mixture. And this flu is going to be able to infect birds and humans. Now, you've probably heard the term emerging viruses before. Emerging viruses are new or previously uncommon illnesses. So of course, the most famous one at this point of time in history is COVID, which is caused by coronavirus. Then there's the Zika virus, there's Middle East Respiratory Syndrome or MERS, there's AIDS, West Nile encephalitis, Ebola, which is hemorrhagic fever, which is fever with bleeding, hemorrhaging. And then there's avian influenza or bird flu. So these are all examples of emerging viruses. Now, what leads to them, there's several different ways that a virus can become an emerging virus. It may extend its range. And, all the traveling that we do all over the world, people traveling from all different places in the world to other places. An example of a virus that became an emerging virus because of this is West Nile, was transported to the United States and took hold in birds and mosquitoes. And then a genetic mutation may occur. So, for example, the influenza strains H5N1, H1N1, and H7N9 were created through the mutation of flu viruses, which only affected animals. And it's necessary to obtain a flu vaccine each year due to the rapidly mutating flu virus. We already know that. But anyway, emerging viruses can either be due to the virus extending its range or a genetic mutation. And then this is just a picture of the globe, and it's showing you different emerging viruses and where they are found in the world. We've got West Nile in the United States now, and coronavirus, and it shows you the year too. So COVID, we know, began in Asia, to our knowledge, and the bird flu, is on here. Ebola in Africa. But anyway, those are emerging viruses and how they get started. Now, viruses are best known for causing infectious diseases in plants and animals. Herpes, HIV, even some cancer is caused by viruses. And antibiotics have no effect on viruses because antibiotics are used for bacteria. There also are disease-causing agents called viroids and prions. Viroids are naked strands of RNA. They don't have a protein capsid. It's just RNA. And they can cause diseases in many plants, particularly crops. Prions are only protein molecules, and they have a contagious tertiary structure. That means their three-dimensional shape has been modified in some way. TSEs are neurodegenerative diseases which destroy nerve tissue in the brain. One type of TSE is mad cow disease. And humans can get that by eating the brains or brain tissue of cattle. The TSE was formerly BSE, or bovine spongiform encephalopathy. So that's the TSE is the form that's found in humans, and the BSE is found in cows. But it basically causes the brain to turn to mush over time. So we have viroids that infect only plants, and prions that cause brain or neurodegenerative diseases. Then we have section 2, prokaryotes. This is just a review. Prokaryotes include the domains bacteria and archaea. So prokaryotes are fully functioning cells. They just don't have a nucleus. They are found in the air, water, and soil, and living on most objects. Louis Pasteur showed that a previously sterilized broth cannot become cloudy with growth unless it's exposed directly to the air. So he showed that there are bacteria in the air. And he came up with the concept of pasteurization and the invention of the pasteurization. He showed that if the flask is open to the air, it's gonna, if the stopper is removed from the flask after it's been boiled, it's still going to become cloudy and show growth after being exposed to the air. And that